This is a neuron. It's made up of dendrites, an axon, a nucleus, a cell body, an axon helic, synaptic terminals to make up a presynaptic cell. Impulses come through the dendrites and flow through the axon from the presynaptic cell to the postsynaptic cell. Now, as the signal travels through the axon, it is sped up by the myelin sheath that surrounds the axon and triggers channels and pumps that lay with the node of Ranvier. These channels and pumps consist of sodium-potassium pumps, a sodium channel, and potassium channel. These channels and pumps are activated, changing the action potential. One channel shows the sodium channel, and another shows the potassium channel and their corresponding ions. A third one shows the sodium-potassium pump. Now moving on to the action potential graph, on the x-axis we have time, and on the y-axis we have membrane potential in millivolts. The graph shows that as the membrane potential reaches a threshold point, it triggers depolarization, which is characterized by sodium intake, and then goes on to repolarize, which is characterized by potassium outflow. It hits a period of hyperpolarization to stop reverse flow of the signal and reaches its resting point again, ready to take on the next impulse. Action potential causes vesicles to fuse with the presynaptic membrane and release their neurotransmitter molecules into the synapse. These molecules bind with the autoreceptors and inhibit subsequent neurotransmitter release. These released molecules bind to postsynaptic receptors, and they are deactivated either by reuptake or enzymatic degradation.
Transmission. We're going to talk about BTX. First, let's illustrate the normal pathway for a neuromuscular junction. In normal transmitter release, the synaptic vesicles containing the neurotransmitter acetylcholine move down the motor neuron towards the synaptic cleft, where the acetylcholine transmitters are then released and bind to their receptors on the muscle cell. Proteins enable the functional transfer of the neurotransmitters. This leads to normal movement of muscles. When BTX is introduced, the light chain acts as a protease and cleaves the proteins that allow the acetylcholine to leave the cell, disrupting the normal release of neurotransmitters. The acetylcholine, which normally transmits the nerve impulse to the muscle cell, is now blocked, causing flaccid paralysis of the muscles, which is characterized by limp and unresponsive muscles.